Now, a few items dealing specifically with kidney histology. First thing we're going to look at is the capsule, the kidney capsule. The kidney capsule has two layers. Has an outer layer. The outer layer is made mainly of typical capsule type connective tissue, a dense, irregular connective tissue capsule with fibroblasts. The second layer, the inner layer of the capsule, is similar, but it has a little less collagen. And in addition to the dense irregular connective tissue and fibroblasts, it has myofibroblasts, a type of a contractile cell that's similar to a fibroblast in its structure. So two layers to the kidney capsule, the outer and the inner layer of the capsule. And here you see the image from your textbook that shows the outer layer and the inner layer of the capsule on the outside of the kidney. Now, when we've talked about the kidney before, we talked about there being a cortex and medulla region in the outer portions and a pelvis in the middle. Where the cortex and medulla come together, we generally say there's a fairly discrete line separating cortex from medulla, but there's a little bit of intergradation, a little bit of a place where there's a inroads of some of the medulla into the cortex out here, and some places where the cortex kind of sinks into the medullary region. All of this material out here where we have the convoluted tubules around that sink down into the medulla out here in the cortex, these are referred to as cortical labyrinths, the cortical labyrinth in the cortex. So some of that sinks down into the medulla a little bit. And on the other side, away from the cortical labyrinths, we have what we call medullary rays. The medulla comes up into the cortex in some places where you see some of the straight tubules, the collecting ducts, extending even up into the cortical region, up in here. So you'll see a lot of straight ducts in this region, many of them being parts of the loop of Henle, as well as straight portions of the proximal and distal tubule. So anyway, these sections of the medulla that reach up into the cortex, called medullary rays, and parts of the cortex that reach down into the medulla. These are your cortical labyrinths. And in this picture, I put two layers to our outer capsule here. So we have the outer layer there and our inner layer here. The outer layer being made of typical capsule material, the dense irregular connective tissue and fibroblasts, the inner layer of the capsule having less collagen and some cells called myofibroblasts in addition to typical fibroblast cells in that inner layer of the kidney capsule. So we have capsule here, we have cortex zone, and then we have medulla there. And although we usually think of those layers as being fairly well separated, some of the medulla comes up into the cortex and some of the cortex sinks down into the medullary region to form these cortical labyrinths. And here's the figure from your textbook that shows where the medulla and cortex kind of come together, where you have an interdigitation and alternating series of medullary rays and cortical labyrinths in that same narrow zone between cortex and medulla. Here we have a drawing of a nephric corpuscle, a kidney corpuscle. That includes the glomerulus structure I've drawn in red here. This tuft of capillaries, this mass of capillaries, forms a circular mass in here. And then around that we have that capsule we call Bowman's capsule. And in there we have that open space where fluid first moves into after moving through the wall of the capillaries. So here's Bowman's capsule, here's glomerulus. The whole thing is a kidney corpuscle or nephric corpuscle. This is the beginning of a nephron unit. So we have an afferent vessel coming in, bringing blood into the glomerulus, and then there's a vessel called an efferent or efferent arteriole leaving the glomerulus there. I'm not going to draw any nuclei in here for the glomerulus right now. We just know that it's lined with endothelium on the inside. Out here on the outside, remember Bowman's capsule had a number of simple squamous cells, and we really just see the nuclei of those simple squamous cells that form the wall of Bowman's capsule out here. So I'll draw in some nice flat nuclei. As we approach the proximal convoluted tubule, where the proximal convoluted tubule leads away from the nephron, our cells change into a nearly cuboidal and then a cuboidal epithelium that lines the proximal convoluted tubule. This end of the renal corpuscle is sometimes referred to as the renal pole or the urinary pole of a nephric corpuscle, and this side over here is called the vascular pole. So you have a vascular and a urinary pole of the structure. So fluid comes in here, circulates through the capillaries, some of the fluid moves out and will eventually be moved into that proximal convoluted tubule. Now, this layer around the outside is sometimes referred to as a parietal layer of Bowman's capsule, very much like what we talked about with the body cavities, how you have an outer parietal layer and then on the organs you have a visceral layer. So the visceral layer of Bowman's capsule consists of some specialized cells that wrap around the outside of the glomerular capillaries. So this tissue doesn't stop here, it reflects on out around there, and I'm going to try to draw this in a bit with some of these special cells. These cells have long foot-like processes that reach out away from them. Again, very much like an octopus kind of appearance to the things. These are podocytes, podocyte cells. So I'll draw some of them around the outside. I'll put some blue nuclei into them. Let's see, there's a nucleus here, nucleus here. So when we talk about these in the laboratory, we'll be looking for podocytes that kind of stick out to the outside of the glomerulus and have obviously rounded nuclei that extend out toward the space of Bowman's capsule. So podocytes, simple squamous cells, cuboidal cells as we go into the proximal convoluted tubule. Let's see, we can label some things here, I guess. Simple squamous cells, proximal convoluted tubule, podocyte, glomerulus, Bowman's capsule. This general region over here would be the vascular pole. This over here would be the renal or urinary pole. We'll just assume this is bringing the blood in as an afferent arteriole. And this is leaving as an efferent or efferent arteriole. The next thing we're going to look at is the filtering membrane between the blood and Bowman's capsule. One other cell type I forgot to mention when going through this renal corpuscle diagram, there's another cell type that kind of works itself in near the base of the renal pole and in between some of the cells out into the glomerulus. Here, these are kind of irregularly shaped, rounded cells out here. Nice ovals, and then they work themselves into the glomerulus here near the renal pole of the kidney. So these special cells, mesangial cells, I'll color them in a little bit here in a minute, put some nuclei in them. Mesangial cells. Have just a few known functions and a few hypothetical functions that are important to, or different roles in the functioning of the kidney. Mesangial cells. Now on to our filtration membrane. In these drawings here we have a couple of views of podocytes. I wanted to show exactly or give some idea of how they fit in the glomerulus. Now I've drawn a very straight capillary here, and of course those in the glomerulus are all twisted around. But this represents the inside. This segment here, what's inside of those capillaries in the glomerulus, just has a lining of endothelial cells, typical endothelial kind of inside to those capillaries in the glomerulus. But on the outside of these cells, you find those podocytes. The podocytes have foot processes. That's where we get the name podocyte. So we have all these extensions that come out from the cells that interdigitate with one another, interlock with one another. Looks like tentacles from an octopus, different octopi coming together, or like lots of spiders around the outside where their legs all interlock with one another on the outside of these capillaries. So that kind of gives a view there. We'll also see a view from the textbook of what that looks like, where they interlock. Now I don't have enough detail in this picture here to show how each of these little arms that come out from each of these podocyte cells will have little legs that interlock again with the neighboring cells all through there. But in this next picture down here, I've drawn larger views of the podocyte. And so here's one podocyte and it has arms, and on the arms that reach out we have these little extensions we call pedicels. These pedicels will interdigitate with pedicels from neighboring cells. So here's a neighboring podocyte and its processes and pedicels that interlock there. This is part of that barrier between the blood and the filtrate. It's what comes out of the blood, out of the plasma, the fluid that filters first through the fenestrated endothelium, then through the basement membrane, and then between the filtration slits, between the pedicels of the podocytes. So that's a little bit of what it looks like. Next, we'll kind of look at a view of the three layers of the filtration membrane: the endothelial fenestrated cells, then the basement membrane outside of that, which I probably should have tried to put in this diagram, and then the podocytes on the very outside of those capillaries in the glomerulus. So in this picture we have the three layers of our filtration membrane of our glomerulus. So on this side would be blood. On this side up here we would have the filtrate, the first stage of urine production out here, that fluid that moved out of the blood, that part of the plasma that was able to get through this barrier and into that space of Bowman's capsule on the outside. So over on this side we have the endothelial cell right here. And it stretches out like this, and I've put in here these little openings here, these little gaps. Those would be tiny pores that actually pass through the entire width of the cells. So each of these little gaps I showed here are actually little pores that are straight tubes that go through from the membrane on one side all the way to the other side of the cell. So these are fenestrations. Right here. So fluids can move through those little pores, and then it will then come in contact with the basement membrane that I've shown in brown here. So that's the next layer of the barrier, the basement membrane. This is often referred to as the glomerular basement membrane, abbreviated GBM. Put that in here, GBM. Glomerular basement membrane. Now your glomerular basement membrane has the, I guess, the most restrictive component of the three layers in terms of that filtration process. Fewer materials are able to pass through the glomerular basement membrane than through either the fenestrations of the endothelium or the little gaps between the podocytes, which we need to mention next here. So these are pedicels of a podocyte up here. Pedicels of a podocyte. And between those little pedicels, between these little foot processes in here, there are some special proteins in here that form a little bit of a grid pattern, interlacing grid pattern. I'll just put some little lines in there to kind of represent those proteins that become established between those foot processes and makes the third layer of the filter here, and we refer to those as filtration slits, podocyte filtration slits. Your book will refer to them as filtration slit diaphragms. So that's that barrier between blood and the space outside in Bowman's capsule there. So all urine that's formed has to start here by passing through these three layers. Small things will not get, large things will not get through there, your larger proteins and so on, but many of your small ions, water of course goes through there. Smaller proteins, very small proteins and peptides will go through there. Sugars, your blood sugar will pass through there, so your blood sugar will have to be reabsorbed in your kidney tubules, as will many of your ions and so on. So anyway, this is your filtration mechanism, the three layers of that filtration membrane in the glomerulus. So here's a photograph from your book showing a podocyte. You can see the main body of the podocyte where it's labeled, and then you can see the arms that reach out from it, And you can see the extensions that interdigitate, those interdigitations between the adjacent feet of the podocyte cell. In this illustration in the book, the filtration membrane is shown with all three parts. This would be the blood plasma side here. It shows the little gaps between the space, or the tiny spaces that go through the endothelium, the fenestrations. The basement membrane is in gray, and then the feet of podocytes, the pedicels, shown out here, the little tiny arrows pointing to that filtration slit membrane between the two feet of the podocytes there. So that's the entire filtration membrane from one side to the other, showing that little magnified part of this electron micrograph in your textbook. This next illustration from your textbook shows between two podocytes here the set of proteins that form that filtration membrane between two podocytes. This is what your book refers to as a filtration slit diaphragm. But anyway, tiny gaps between proteins, interlaced proteins between the interlaced feet or pedicels of the podocyte here. What's labeled GBM is your glomerular basement membrane. That's basement membrane from the endothelial cell. And then you can see the fenestrations in the endothelial cell there below. Now we're going to talk a little bit about the functions, possible functions of the mesangial cells that are found in the base of the nephric corpuscle. The first one is cleaning of the glomerular basement membrane, here again abbreviated GBM, cleaning of the glomerular basement membrane. So they may have a phagocytic role where they get in there and between things and can remove whatever may be clogging the filtration slits. Structural support next. It's thought that there are places where the podocytes don't exactly fit against maybe some of the basement membrane or other structures. And anyway, some possible structural role there. Next, number 3, repair-inducing chemicals. It's thought that if some kind of damage is occurring to the glomerulus, that there are some chemicals here that could cause or help those cells repair themselves in there. So repair-inducing chemicals released from the mesangial cells. And then the last one here, contractile properties that might be important in controlling the blood flow through the glomerulus, through those small vessels and passages.